Welcome back. Sexual reproduction in flowering plants is packed with precise vocabulary and processes that show up constantly in NEET, from pollen formation to the unique process or double fertilization. In this video, we'll walk through the male and female reproductive structures, pollination, the remarkable process of double fertilization, and how the fertilized flower develops into a fruit and seed. Let's get started. The flower is the reproductive structure of angiosperms. The reproductive part is the stamen, made up of a filament supporting an anther, where pollen grains are produced. Inside the anther, specialized cells called microspore mother cells undergo meiosis to produce 4 haploid microspores, each of which develops into a pollen grain. A mature pollen grain typically contains 2 cells, a large vegetative cell, which will later form the pollen tube, and a smaller generative cell, which divides to produce 2 non-motile male gametes, meaning sperm cells without any means of independent movement, since plant sperm can't swim to the egg on their own, unlike in many animals. The pollen grain wall has 2 layers, an outer exene, made of an extremely tough and resistant material called sporopollenin, which is why pollen grains fossilize so well, and an inner intine, made of cellulose and pectin. The female reproductive part is the pistil, or carpel, consisting of the stigma, where pollen lands, a style connecting it to the ovary at the base, which contains 1 or more ovules. Inside each ovule, the megaspore mother cell undergoes meiosis, producing 4 haploid megaspores, but typically only 1 survives, and it develops into the embryo sac, the female gametophyte, through 3 rounds of mitotic division. The mature embryo sac in the common polygonum type contains 8 nuclei arranged in 7 cells. At the end nearest the opening, called the micropyle, sit the egg cell flanked by 2 synergids, together called the egg apparatus. At the opposite end sit 3 antipodal cells, and in the center sits 1 large central cell containing 2 polar nuclei. This precise arrangement, and the specific role of each cell, is exactly the kind of detail NEET loves to test. Pollination is the transfer of pollen grains from the anther to the stigma, and it's classified based on the source of the pollen. Autogamy is self-pollination within the same flower. Geitonogamy is transfer between 2 different flowers on the same plant, which is genetically still considered self-pollination, since the genetic source is identical. Xenogamy is transfer between flowers on 2 genetically different plants, the only type that introduces genuinely new genetic variation. Plants have evolved a wide variety of pollinating agents. Many rely on wind, called anemophily, water, called hydrophily, or animals, especially insects, called entomophily, with flowers evolving specific colors, scents, and nectar rewards to attract their particular pollinators. Many plants have also evolved mechanisms specifically to prevent self-pollination and promote cross-pollination, since cross-pollination generally produces more genetically diverse, and often more vigorous, offspring. After landing on the stigma, a pollen grain germinates, growing a pollen tube down through the style toward the ovule, carrying the 2 male gametes inside it. Once the pollen tube reaches the embryo sac, it releases both male gametes, and this is where the truly unique process of double fertilization occurs, found only in angiosperms. The first male gamete fuses with the egg cell, forming a diploid zygote, which will eventually develop into the embryo. This is called syngamy, ordinary fertilization. The second male gamete fuses with the 2 polar nuclei in the central cell, forming a triploid, or 3N, cell called the primary endosperm nucleus. This is called triple fusion, and it develops into the endosperm, the nutritive tissue that nourishes the developing embryo. Because 2 separate fusion events happen using the 2 male gametes delivered by a single pollen tube, this entire process is called double fertilization, and it's a defining feature that distinguishes angiosperms from gymnosperms and all other plant groups. After double fertilization, the flower undergoes a coordinated transformation. The zygote develops into the embryo, going through globular, heart-shaped, and torpedo stages before maturing into a fully formed embryo with a plumule, the future shoot, and a radicle, the future root. The primary endosperm nucleus divides repeatedly to form the endosperm tissue, which stores nutrients, and in some seeds like beans, this endosperm is entirely consumed by the growing embryo before the seed matures, while in others, like cereal grains, a substantial endosperm persists in the mature seed. The ovule as a whole develops into the seed, with integuments hardening into the protective seed coat. Meanwhile, the ovary wall develops into the pericarp, the fruit wall, and the entire ovary matures into the fruit, which protects the developing seeds and often aids in their dispersal. In some species, fruit can develop without fertilization at all, a phenomenon called parthenocarpy, producing the seedless fruits, like some bananas and grapes, that we eat. Let's recap. The stamen produces pollen grains carrying two male gametes, while the pistil's ovule develops an 8-nucleate embryo sac with an egg cell, two synergids, 3 antipodal cells, and a central cell with 2 polar nuclei. Pollination can be self-pollination, autogamy or geitonogamy, or true cross-pollination, xenogamy, aided by wind, water, or animals. Double fertilization, unique to angiosperms, involves syngamy forming the diploid zygote, and triple fusion forming the triploid endosperm nucleus. And after fertilization, the zygote becomes the embryo, the ovule becomes the seed, and the ovary becomes the fruit, sometimes even without fertilization at all in the case of parthenocarpy. That's sexual reproduction in flowering plants. The embryo sac structure and double fertilization process are extremely high yield for NEET, so make sure you know them cold. See you in the next video.